This fitness video is going to focus on egg production, which is called oogenesis. And again, you're gonna see some similarities between spermatogenesis, but then also some big differences. So, egg production does produce haploid gametes, and it is by the means of meiosis, but it is very cyclic. So it only releases one egg each month, and that is accompanied by cyclic changes in the hormones and then also in the structure of the ovaries and the uterus. And so the uterine changes that we see is the result of our monthly menstrual flow. So let's focus on how oogenesis starts in embryonic development. So the germ cells are going to arise from the yolk sac during the embryonic development, and we are going to see those same ridges that we talked about in the male video. And then those ridges are going to differentiate into oogonia, and they will multiply until the 5th month. They're going to multiply up until 5 to 6 million oogonia, and they are going to transform into primary oocytes. And this is early meiosis 1. And so here, unlike in sperm, the meiosis cell divisions are going to be broken up. So it's not kind of one smooth process like it was for the sperm cells. This is a little bit different. So the oocytes are in early meiosis 1, and then most of them degenerate by the time that the girl is born. We call them eggs or ovum, any stage from the primary oocyte to the time of fertilization. At puberty, there are 400,000 oocytes left, and that's a lifetime supply because a normal female would ovulate 480 times. Ugh, sounds like a lot when you say it like that, but so that is plenty of oocytes for a lifetime supply of ovulation. So the egg development resumes in adolescence when FSH stimulates the monthly cohorts of oocytes to finish meiosis 1. And so then from there, meiosis 1 means that that's the first cell division of meiosis. And so that first cell division, the one cell divides into 2. So each oocyte divides into 2 haploid daughter cells. They are unequal size, so this is already a difference between spermatogenesis. They are very different. One is big and one is very little. The reason for this is to have an egg with as much cytoplasm as possible. So when you see the end of meiosis 1, you'll see one big cell and one little tiny one. That's called a polar body. So if the egg is fertilized, it has to divide repeatedly soon and produce numerous daughter cells. So that's why as much cytoplasm as possible is a benefit. And then from there, we call it the secondary oocyte. So that's the large daughter cell that is the product of meiosis 1. And I'm hoping for a picture soon. Actually, I'm going to jump to a picture. There we go. We're on the left-hand side in the blue. And so you can see at the end of meiosis 1, we have one really big cell and one little tiny cell. And so the big cell is called the secondary oocyte, and then the little one is called the first polar body. So let's go back and talk about the first polar body. Sometimes it undergoes meiosis too, but really we don't need to worry about that because ultimately it disintegrates and dies. It's the mean of discarding the extra set of haploid chromosomes. So that secondary oocyte still has to only be haploid. I shouldn't say only. Has to be haploid because when it gets fertilized by the sperm, that's when it's going to go back to a diploid number of chromosomes. But so it has to be haploid. So the secondary oocyte goes as far as metaphase 2. Now this feels really weird because we're going to start the second part of meiosis but not finish it. And it arrests until after ovulation. So if it's not fertilized, it dies and never finishes meiosis. But if that secondary oocyte is fertilized, then it completes meiosis 2 and it casts off a second polar body. And then again, the haploid secondary oocyte would meet with the haploid sperm, and that is how sometimes it's called restoring the diploid number or going back to a diploid number of chromosomes. And then, again, our magic number is 46. So we're just looking at the left-hand side. The right-hand side is the development of the follicle. We'll get to that. But the left-hand side is kind of where we're at right now. And so the secondary oocyte, if it's fertilized, it is going to then finish up meiosis 2, cast off a second polar body, and then the oocyte that was fertilized and the sperm, those are together called a zygote. That's what an egg and a sperm are called when they have fertilized each other. And notice you see 2N. That means a diploid number, a full set of chromosomes. So this is kind of a microscopic look at what the follicles look like. And this is going to talk about primordial, primary, and secondary follicles. This is just showing that they're kind of commingled. We're going to talk about which follicles are actually producing oocytes. We're going to focus on secondary follicles here pretty soon. So the development of the follicles has a ridiculous name, folliculogenesis, and that is the development of the follicle around the egg, and the egg is undergoing oogenesis. So we're going to talk about the different types of follicles, kind of the different stages in this developmental process. So the original primordial follicles, that's what we saw around the outer edge of the ovary. And so they're where the primary oocytes are. They are in early meiosis. And so they are surrounded by a single layer of squamous follicular cells, and they connect to the oocyte and to be able to pass nutrients and signals. They are in the cortex of the ovary. And then they wait 13 to 50 years before they develop further. And they are each developing or finish developing at different points. Remember, at the end, each follicle is releasing one egg every month. So they all have different kind of times when they are done developing. But adult ovary has 90 to 95% of the primordial follicles. Then the next stage is primary follicles. They have larger oocytes, but the cells still form a single layer. Then the secondary follicles, again, still larger oocytes, but the cells are now stratified. And so those stratified cells are where we're going to start to see kind of this layer around the oocyte and be able to see the follicle develop. Again, I'm not really picky about you knowing about granulosa cells or these different zones. I'm really not that picky. We've got a lot going on this week. So the tertiary follicle, they start to secrete this fluid. They make little pools in the wall, and that is what defines the tertiary follicle. But as they enlarge, the pools merge, and they make a fluid-filled cavity called the antrum. And so I want to get to a picture of that, and we'll go back to this. There's a picture, but this is a microscope picture, so it's they all kind of look the same. I'm actually going to backtrack to, not that one. Oh my gosh, I got to backtrack really far to find it. Okay, so this mature follicle, we're almost there. We're here in this secondary follicle, and these little fluid-filled cavities is what we're talking about. And so you can see these stratified layers of cells and how that looks very different than the primary follicle. I feel like that picture probably should have been labeled tertiary. Oop, I went too far. Okay, so those are the tertiary follicles. And this talks about how we have a mound of granulosa cells on one side, and that covers the oocyte and secures it to the wall of the follicle. Basically, we have a layer of cells that makes a barrier around the egg, similar function to the blood testes barrier. Makes two layers. Again, I feel like there's a lot going on here, and I just kind of want you to know the overview. But the mature follicles, normally one follicle from each month's cohort becomes mature, destined to ovulate, and the remainder degenerate. So only one follicle is going to ovulate each month. and then the rest are going to degenerate.